Good morning, students. Today we will discuss biochar and other biological approaches used in food byproduct and waste management. Modern waste management focuses not only on disposal but also on converting waste into valuable products. Biochar production, insect-mediated bioconversion, vermicomposting, biodrying and ensiling are sustainable technologies that help transform organic waste into useful resources. In this session, we will understand these approaches, their mechanisms, advantages, and practical applications. Let us begin with the concept of biochar. Biochar is a porous carbon-rich material produced through the thermal decomposition of biomass in the absence of oxygen. This process is known as pyrolysis. The slide also describes biochar as a charcoal-like substance produced from agricultural and forestry wastes. Because of its high carbon content and stable structure, biochar resists decomposition and remains in the environment for long periods. This property makes biochar important for waste valorization and carbon sequestration. Let us examine the important characteristics of biochar. The first characteristic is its highly porous and fine-grained structure. This provides a large surface area for adsorption and nutrient retention. Another characteristic is its hydrophobic nature, meaning it tends to repel water under certain conditions. Biochar also contains small aromatic carbon structures that contribute to its stability. An important point is that biochar composition varies depending on the feedstock used and the pyrolysis conditions applied. Therefore, biochar produced from different materials may have different properties and applications. This diagram illustrates the overall biochar production process. On the left side, we can see various feedstocks such as manure, organic wastes, crop residues, energy crops, and biomass materials. These feedstocks enter a pyrolysis unit where thermal decomposition occurs without oxygen. The diagram shows that part of the carbon is converted into biofuels such as bio-oil and hydrogen. These products can be used for transportation, energy generation, industrial applications, and co-product manufacturing. Another portion of the carbon is returned to the soil as biochar. The diagram also highlights that bioenergy products such as syngas and bio-oils are generated alongside biochar, making the process both environmentally and economically beneficial. This diagram presents different biomass feedstocks used for biochar production. These feedstocks are broadly classified into lignocellulosic biomass and non-lignocellulosic biomass. Lignocellulosic biomass includes agricultural residues such as rice husk, straw, corn stover, bagasse, wood chips, sawdust, bark, energy crops, and municipal solid waste. These materials are rich in cellulose, hemicellulose, and lignin. Non-lignocellulosic biomass includes sewage sludge, animal waste, algal biomass, fur waste, and skeletal waste. These materials generally contain higher levels of nitrogen, phosphorus, sulfur, and heavy metals. Therefore, careful management is often required before their use in biochar production. This flow diagram explains the pyrolysis pathway. The process begins with biomass collection and material preparation. The prepared biomass is then subjected to pyrolysis. Depending on operating conditions, pyrolysis may be categorized as slow pyrolysis, fast pyrolysis, or flash pyrolysis. The products obtained include biochar, bio-oil, and fuel gas. The diagram clearly demonstrates that changing the pyrolysis conditions influence the quantity and type of products generated. Thus, pyrolysis can be optimized depending on whether the objective is maximum biochar production or maximum energy recovery. This diagram provides a detailed comparison of different pyrolysis technologies. Slow pyrolysis generally produces the highest biochar yield, typically between 30 to 60%. Its major advantage is high char production, although the process requires longer residence times. Fast pyrolysis produces lower biochar yield but higher bio-oil yields. Flash carbonization offers shorter residence times and lower operating temperatures but requires higher pressure. Microwave-assisted carbonization provides uniform heating and improved product quality, although temperature control can be challenging. The lower portion of the diagram also shows biomass decomposition stages, including drying, primary pyrolysis, and secondary pyrolysis, which ultimately produce biochar, bio-oil, and biogas. This table provides a detailed comparison of slow, intermediate, fast, flash, and microwave-assisted pyrolysis. The comparison includes temperature, heating rate, residence time, particle size requirements, moisture content, product distribution, and biochar yield. Slow pyrolysis operates at relatively lower heating rates and provides the highest biochar yield. Fast and flash pyrolysis operate at higher temperatures and heating rates, producing larger quantities of bio-oil. Microwave-assisted carbonization uses microwave energy for rapid and uniform biomass heating. The table clearly demonstrates how operating conditions influence product characteristics and process efficiency. Let us now discuss the applications and advantages of biochar. Biochar is considered a cost-effective alternative to activated carbon for contaminant removal. Its large surface area, microporous structure, and high adsorption capacity make it highly efficient in pollutant capture. Biochar also possesses ion exchange properties that help remove both organic and inorganic contaminants. Its performance depends on the feedstock source and pyrolysis conditions. Major applications include wastewater treatment, soil improvement, composting, carbon sequestration, environmental remediation, and energy storage. These diverse applications make biochar an important product in sustainable waste management. Another important biological approach is insect-mediated bioconversion using black soldier fly larvae, commonly called BSFL. These larvae act as efficient biological reactors for processing high-moisture organic wastes such as food waste, fruit waste, vegetable waste, and market residues. The larvae rapidly consume and digest organic matter, reducing waste volume and mass by approximately 50 to 60% within 10 to 14 days. The harvested insect biomass contains up to 40% protein and 30% lipids. This makes it valuable as an alternative feed source for aquaculture and poultry industries. The remaining residue, called frass, serves as a nutrient-rich soil amendment. This process perfectly illustrates the concept of circular economy. Another sustainable technology is vermicomposting. Unlike conventional composting, vermicomposting is a low-temperature biological process involving earthworms and microorganisms. Commonly used earthworm species are Eisenia fetida, known as the red wiggler, and Eudralis eugenia, known as the African nightcrawler. The worms mechanically grind and fragment organic waste in their digestive systems. This increases surface area and promotes microbial activity. Enzymes such as cellulases, proteases, and lipases further degrade organic matter. The final product, called vermicast, contains higher levels of available nitrogen, phosphorus, potassium, and plant growth regulators compared to conventional compost. Therefore, vermicompost is highly valued as an organic soil conditioner. Let us now understand biological drying, also known as biodrying. Biodrying is commonly used in mechanical biological treatment plants for mixed waste management. The process utilizes metabolic heat generated during aerobic degradation of biodegradable waste. Instead of maximizing decomposition, the primary objective is rapid moisture removal. Forced aeration is used to accelerate drying. Typically, moisture content is reduced from more than 50% to below 15% within 7 to 15 days. This significantly increases the calorific value of the waste material. The dried waste can then be converted into refuse-derived fuel, or RDF, for energy generation. Therefore, biodrying serves as an important pre-treatment step in waste-to-energy systems. The final biological approach discussed here is ensiling of organic solid waste. Ensiling is mainly applied to homogeneous food industry wastes such as fruit peels, potato pulp, brewers' grains, and fish processing waste. The process involves compacting the material to exclude oxygen and create anaerobic conditions. Under these conditions, lactic acid bacteria multiply and ferment soluble carbohydrates. The primary product formed is lactic acid. As lactic acid accumulates, the pH drops below 4.5. This acidic environment prevents the growth of spoilage and pathogenic microorganisms. As a result, the waste becomes stabilized and preserved without requiring energy-intensive drying. The preserved material can later be used as high-quality animal feed. Thus, ensiling provides an economical and effective preservation strategy. Thank you.